Okay. Hello and greetings everyone. Let me know if you can hear me.
Yes.
Okay, so perhaps some are struggling with their network and they are joining through YouTube. So because we're streaming via YouTube for most persons that are struggling with their network and for those who are still coming back from work and or that they can watch the recorded version. Okay, so thank you guys. I welcome you to class 11 of the Afrolongevity Educational Program Cohort 3. And today we'll be having an interesting conversation around development and application of aging clocks. And today we have uh, Dr. David Mayer as our instructor. He is a passionate bioinformatician. He is so interested in what is happening in Africa, doing his best. In fact, we are the one that is slowing him down, and he's willing to share his wealth of knowledge and then to support those who are really interested in developing or bridging the longevity divides from wherever you are sitting and your area of functioning. So he's also interested in genome instability and aging research. Um, so in ensuring that he makes meaningful contribution to the field of biogerontology. So thank you so much, Dr. Mayer, for making up time to be here and always being available in helping us to learn and to do the best we can in contributing in bridging the longevity divides in Africa. I yield. Thank you.
Thank you for the nice introduction, and also thank you for inviting me for a second time for this. I really like being part of this. I think it's really amazing what you're building at the moment. Today I will give you introduction to the development and application of aging clocks. And to start off with, you know that everyone of us ages. And this can happen at different speeds. Some of us age faster or slower than others. And the question is, of course, why? And to be able to understand the why, we first need to be able to measure this difference in the biological age. So how can we do so? First of all, we of course need to understand what the difference is between biological and chronological age. Chronological age is your calendar year, but as Benjamin here already pointed out over 60 years ago, is that one man may be 60 years old while another might be 60 years young. So maybe one 60-year-old person is still running marathons, whereas another is already frail and close to death. So the question really is, how old is old? And the calendar does not hold the key to this question. There's something else. And in this publication, Benjamin proposed a way to measure the biological component. We call it functional age. And he had this questionnaire. Questions are what you maybe know from your doctors already. So it's quite usual, like this diet, eating habits, smoking, how well you sleep, and so on. So nothing unusual, and something that is still getting used today, of course. And this in the end then gives you a score, which then tells you how functional or biological old you are. So this was the first way to test your biological age. And of course, this gives you some insights, but it doesn't give you any mechanism. Or you don't know why it's happening. A few years later, people became more and more interested in this biological functional component, and especially this group that was interested in irradiation and whether irradiation might affect aging. So over the 40s and 50s, people have irradiated animals and have seen that it does not only lead to cancer, but also that it leads to an accelerated aging phenotype due to most probably DNA damage. And no, the people were interested in whether this might also happen in humans. And in Hiroshima, Japan, we of course have a population that underwent potentially high doses of irradiation after the bombing of Hiroshima. So this group now had a look at people that were living close by or distant enough to not have any irradiation, and were looking at whether there's any difference in the biological component. So they grouped these individuals into 4 groups. Group 1 were closest to the irradiation zone, whereas group 4 was under no irradiation whatsoever. And because at that point there was no agent clock available, what they did is they asked nurses to estimate the chronological age. And you can see on average for the males and females here separately, these nurses are highly accurately able to predict the age actually. So it's less than 1 year difference. I couldn't do that. But they were not interested in whether the people that were under high irradiation when the bombing happened, whether they might look older than people that were under no radiation. But you can see there's no clear pattern. Males are generally predicted younger than females, but there's no pattern with irradiation. So it was a negative result. But they came back sometime later with some other measures and wanted to see more of biomarkers of aging. So not estimates from nurses, but actual biomarkers. And here you will, of course, always think some biomarkers that tend to go up with age, some that tend to go down with age. But every single biomarker doesn't give you that accurate results, or also not that big of an insight. So over the years, and starting already, of course, several decades ago, people have investigated machine learning approaches, often as simple as linear regressions, that combine many of these biomarkers into something we call aging clocks that highly accurately can predict the chronological or know subsequently also the biological age of organisms. And this group tried the first real aging clock, I would say, and they measured these different biomarkers from hair graying to skin elasticity, grip strength, but also blood pressure and so on, for different individuals. They see that there is indeed a strong correlation or anti-correlation with age. And based on these, they built the first aging clock based on a linear model that was able to predict what they call the physiological age of males and females to a decent accuracy. You can see it's quite flat until the age of 35, but afterwards, the accuracy really kicks in. However, and this they don't show in a graph, but they had a negative result here as well. They also investigated, again, the Hiroshima population, people that were nearby the irradiation and those that did not receive any radiation. And there was no difference in the predicted aging score. So it was again a negative result. So the biological component does not seem to be predictable. And this also then led to a slowing down of the field. People were not investigating it so much, and people were even discussing whether it's even possible to predict the biological age. Maybe it's just not possible, or you would need to do this for every single cell in your body, because maybe there is not just one common biological age. And that then really took a lot of time until epigenetics came around to really kickstart the field again. Just a brief reminder what epigenetics is. In every single cell of your body, you of course have the same DNA, but a liver cell, a neuron cell, a muscle cell all have different functions. And this functionality, this difference is of course regulated in some way, and this regulation is happening with epigenetics. The most simple way, and also what I will discuss later, is DNA methylation. So these are small modifications directly on the DNA. Usually, if they are at the promoter region, they are repressing the gene. So in a liver cell, you don't want neuronal genes active, so they are usually repressed with DNA methylation, for example. But then you also have other modifications on the histones where the DNA is wrapping around. It's getting quite complicated, and of course, a lot of it still is not known, but we will focus here on the DNA methylation part. And why is this now interesting for the aging clock field? This came from a paper, I think somewhat around 20 years ago or so, where people were interested in monozygotic twins. Because monozygotic twins, they of course share the DNA. They are essentially identical at birth or during embryogenesis. But with time, with age, these individuals of course diverge. They get more and more distant from each other. And the question is, how could it be if the genes are completely identical? And the epigenome was, of course, a candidate, because that is happening in every single cell differently. Every single cell has a different epigenome, and that is also regulated dynamically. So they wanted to check, could the epigenome explain this? And indeed, this is what they found. Epigenetic differences arise during the lifetime of monozygotic twins. So there is something happening with the epigenome with aging. Also different in different individuals, even in different twins. And this, of course, now was interesting for the aging field, because now we have here a different biomarker, the DNA methylation, which seems to change with age. So it didn't take long for the first epigenetic predictor of age from Bokland et al. This was a small study, but they showed that in principle, you can build a predictor based on the epigenome, on DNA methylation, that is fairly decently able to predict the chronological age. People were still arguing this might not be relevant because, as I've just told you, every single cell type in your body has a different epigenetic setup. A liver cell has different genes active or inactive than a muscle cell or a brain cell. So in theory, every single cell type should age differently. There shouldn't be a common signal. Every cell has different genes active and inactive. But to the surprise of everyone, Steve Horvath, a few years later in 2013, showed in this landmark publication that it is indeed possible. He showed that he can use one single clock that is able to predict the chronological age of an individual irrespective of his cell type. He just did this by accumulating all the data that there's and just build one gigantic regression model on all the CPG sites over all the training data that he could get his hands on. And he showed that it is indeed possible. There is a common signal in the DNA methylation data. But now this is chronological age. But the hypothesis is here now, and this is also in Horvath's clock paper, is that the prediction error contains biological information. So if you are chronologically speaking 50 years old, and your prediction is 50 years, you are an average individual. But if you are predicted younger, let's say 40 years, you are aging slower. Whereas if you're predicted 60 years, you might be a fast ager. And indeed, it was then shown that aging clocks are associated with mortality. Here's just one example. This is PhenoAge. This is a second generation aging clock, but it's the same for other clocks, just that some clocks behave better than others. You can see that individuals that were predicted younger than their age, so 10 years younger, they actually survive longer than those that are predicted older than 10 years. So there's a pretty big difference here. So on the y-axis, you can see the percentage survival. On the x-axis is the chronological age. And you can see there's a pretty big gap in survival in between those populations that are predicted older or younger. So there is indeed biological information in these aging clock predictions. It is possible to predict mortality to some degree. However, there are still several problems in the field. One of the biggest problems is that almost all data is cross-sectional, and cross-sectional data does not allow proper validation or identification of causal effects. So what is cross-sectional? This is just if you have one cohort and you measure every single individual just one time, you might have a range of chronological age. So maybe your cohort is big, you have maybe a period from 20 to 100 years, but still every individual is just measured once. The problem is, of course, that every individual might have a different baseline, it might have different aging rates, it might be stressed at a certain time, and so on. So you cannot really infer much. You can still build these aging clocks, even those that predict mortality in cohorts, but on the individual level, it's really still really hard because every individual might be different. And this is something we cannot really investigate without cross-sectional data. For this, we would need longitudinal data. So we have the same individuals measured multiple times, ideally, of course, over a longer time frame, so that we really can have individual aging trajectories to then build way better aging clocks that can predict these trajectories and individual aging rates. The problem here is just that the data is not there. There's not much longitudinal data, but this is something where the field is, of course, going towards too. So this is the goal, to have longitudinal data, but this, of course, needs long-term planning because you need a cohort and you need really to make sure that you can measure these every year or every other year. Another problem is that aging clocks, especially also average aging clocks, are cell type composition confounded. What do I mean by that? If we look at blood cells, for example, these are known to change with age. Your blood, your PBMCs, the blood cells are, of course, you have different subtypes of the CD4 plus cells, there's the CD8 cells, you have memory cells, natural killer cells, monocytes, and so on. And depending on your age, these might change quite heavily. So for example, here the naive CD8 T cells, you can see they're quite common in younger people, but they completely drop the older you are. And then there might be other cell types that increase their fraction. Now, I've just told you that every single cell type has their own epigenetic setup, because every cell type needs their own genes to be active or inactive. So the DNA methylation setup is different in these cells. And now, of course, you might have the fact that within each cell, maybe there's no change whatsoever. Maybe the CD8 cells and the CD4 cells, there's no aging change. They're completely stable. But just because now you have less CD8 cells with age, you suddenly see, if you merge them together in blood, so you just have a bulk sample, that there is a strong aging trend. And then, of course, you will see that especially DNA methylation sites that are specific to CD8 T cells, or those genes that are active there. But it's not the cells themselves that change, it's just the composition that changes. So it's confounded. You have different information. It's still useful. Yeah, you can still predict mortality, but the underlying interpretation is completely different. And this is also then visible in several data sets. Yeah, we do see a stress-induced cell type composition change, and this can conform predictions. Here on the left side, for example, is a study from an emergency hip surgery, and they measured a DNA methylation clock. Directly before the surgery, directly after the surgery, time point 2, or several weeks afterwards, when everything was healed again. And you can see that directly after the surgery, there's a significant increase in the predicted age, almost 10 years, although there was not much time between them. It's just that after the surgery, you have inflammation, you have a change in cell type composition, and this is what these clocks measure. And this is, of course, the case because with aging, you have inflammation, right? You have an increase in inflammation, low-grade inflammation. And this is what these clocks then see. So they do see with age, inflammation is going up, and they learn to associate aging with inflammation. And this is why now if you have inflammation, suddenly your clock is pretty good older. This is especially also visible in acute infections. So here, for example, is a transcriptomic clock paper with viral infections. The same for influenza or COVID. And you can see in the acute sick phase, these individuals are on average 25 years older. And then this decreases again back to more or less normal levels once they are healthy again. And this is, of course, different to interpret because ideally your biological age should not be affected so much by a simple infection, right? Because infection that can go up and down, but you're healthy again, and afterwards you're back to normal levels. But this is not what I would call the real underlying biological age of an individual. This is still important. This is still measuring mortality and so on, but the interpretation shifts. And even the circadian rhythm can impact the predictions. So you can see it. This is a really nice paper from 2 years ago. The same individual sampled multiple times during the day, and you can see the age of this individual really nicely fluctuating. So it starts at maybe 54 or 53 years. It peaks at more than 60 in this one sample, and then it goes down again. And this follows nicely the circadian rhythm. And of course, this makes sense because during the day, your cell type composition also changes. In the morning, when you wake up, stress hormones rise to make you wake, the cell type composition changes. And in the night, when you get to rest again, it changes again. And this is exactly what is reflected here as well. It's, of course, also a problem because there's so much variability just within one day from almost 10 years difference in the prediction due to these potential confounders. And this is, of course, something that the field wants to improve on. There have not been improvements, but it's still something that is not completely solved. Another problem is that many aging clocks can be found. So there's still another question, how can we interpret them? Because if you have an aging clock on DNA methylation sites or genes, usually the first thing is to ask, is there anything specific about this DNA methylation site? Is there anything specific about the genes? Would it be possible to maybe slow down aging if we do something about those genes? But now this nice paper from 5 years ago showed, for DNA methylation at least, that you can build multiple aging clocks from the same data. So if you build an aging clock, you can predict age. And now you take out those CPG sites that are important for predicting the age, and you just do again. So you just retrain your aging clock. Then you find a clock that is almost exactly as accurate as the previous one. They found up to 20% of sites can be actually used for aging clocks. So there's a lot of redundancy. And it's not like this specific CPG site, this specific gene is really driving aging. They are more downstream readouts. This is also then a question that sparked our interest. And we were wondering, what actually is the underlying signal of aging clocks? And how can the prediction really be explained? And this is something we published 2 years ago as our own research. And here we wanted to ask how much a stochastic process could explain. Because with aging, we assume that there's a lot of damage happening. Yet you have DNA damage, you have mutations and so on, and these are random events. But then again, aging clocks are almost deterministic. You can predict the age of an individual with really high accuracy by now. The question is, how does this fit actually together? How can a random process lead to something that is highly predictable? And also, why do you often find similar genes enriched? So often you find certain pathways, like developmental pathways. How could this be? Or do you need maybe a program of aging? Maybe these damages are not relevant. Maybe it's really just a program. And here we started with a random process simulation, stochastic process. In the beginning here, there's no biological data. This comes later. This is just to show you the principle. So we start with the ground state, for example, just 4 values between 0 and 1, doesn't matter here. And then we just simulate a random process. This can be visualized by such a golden board. You probably have seen it before. You throw sand or stones on top and it trickles down. And in the end, you have something that resembles a normal distribution. So it's a random values. And now we just choose small random values, equally likely to be positive or negative, and we just add them to the ground state. It's just random values that we add. And doing this once, we just define to be one time step. So the ground state is H0, and adding noise once is H1. And now we can, of course, do this not only one time, but let's say up to 100 times. We just add more and more noise. So H1 is still somewhat similar to the ground state. H100 is quite different because we added noise so often.
No. Yeah.
So is there a question or? Okay, probably not. So we just have more and more noise in the data. And now we use this data set to train a predictor. This is just a linear regression model. And we just try to predict how often we added noise. So how the simulated age, so often we added noise to the data. And now we, of course, need to test it. So we do exactly the same ground state. We add noise. We have independent test data. And now we want to see whether the model learned anything. Can we predict how often we added noise to the data? And in the beginning, we did not see anything. The prediction is completely flat. It's not possible to predict anything. If you just add more and more noise, more and more stochastic variation without any limits, you cannot predict anything. This also should not surprise because noise, by definition, should not have a pattern. So you cannot learn anything. There's no pattern in the data. But then we thought this is not how biological data behaves. In biology, you usually have some kind of constraints. DNA methylation, for example, within a single cell is binary. It's 0 or 1, not methylated, methylated. if you have a bulk set, a bulk-based set, so the average of thousands of cells, it's between 0 to 100%. Either every single cell is methylated 100% or unmethylated 0%, or something in between, of course. But it does not make sense to have minus 10% or 110% methylation. So you have constraints, you have limitations. And now if we do exactly the same as I showed you before, we just add noise to the data, but we have limits. We say it cannot go below 0, it cannot go above 1. Then suddenly we see that we can build accurate predictions. So we can predict how often we add noise to data. So this just shows, in principle, it's possible. This is artificial data, it's not biological data. But it shows that in principle, you can predict how often noise was added to data. Now the question was, of course, how about biological data? And the first question we ask is, how would an age predictor behave, like an epigenetic clock from Steve Horvath, on data with accumulating stochastic variation? So if we just add noise. So we do the same as before, but now we start with a biological sample. With a biological sample, maybe a 20-year-old human. If we use the epigenetic clock, it should be predicted about 20 years. But now we add noise, more and more noise. What would happen to the predictions? Would they behave randomly, or would they show any pattern? And to our surprise, we did see a really clear linear correlation. So the x-axis here is simulated age, so how much noise we added, up to 100 times. And the y-axis is the predicted age with Horvath epigenetic clock, the one I just showed you. And you can see it's a clear linear correlation. The more noise we add, the older the predicted age. This really surprised me. I didn't think this would happen. And this is the same for every aging clock we tested. Every first or second generation aging clock, also for transcriptomic aging clocks. It's always correlated. And now we're wondering, if this works, can we do the reverse? Can we use a simulated data set and build our own aging clock? So again, it's the same as before. We start with a biological sample, maybe a 20-year-old human DNA methylation sample. We add noise. And then we train a predictor that tries to predict the simulated age. So the same as I showed you before. But now we actually tested on biological test data. So we now have independent data from humans, maybe from 20 to 80 years or so. And we want to see, could this model that just learned on noise predict anything? And the really surprising thing is that this works really well. So here now on the x-axis is the chronological age of human samples. Every dot is an independent human blood DNA methylation sample. And on the y-axis is the predicted age with a clock that just use one single biological sample, and everything else is just noise that we add. So this really shows what these aging clocks learn and need is noise. You just need noise in your data, because with age, you have more and more noise. So that was human. But we went a step further. I wanted to look at pan-mammalian. There was a recent publication from Steve Horvath again, who beautifully showed that he can build one single aging clock to predict the age of all these different animals to a really high accuracy. So you can see it's going from the mouse to humans, but all the way to whales. And with these lines here on the circle, it shows you how accurate the clock is. So the further outside, the more accurate. You can see it's on average, I think above 0.8 or 9 or something like this. So it's really highly accurate. And now we wondered, even across different mammals, can we do the same? So here in this case, how they train the clock is they have thousands or even 10,000 different samples from all these different animals. They train one clock, and then they predict one species. So maybe they take the humans out, they train on everything else, and then they predict humans. And this works really well, as you can see here. What we now did is we just took one single sample from, in the beginning, from the bottlenose dolphin, just one biological sample. We just added noise, and we tested on all the other samples now independently. And we saw that most samples, most species, are actually highly accurately predictable. So you can see here again with this spooky line here around the circle, then on average, it's above 0.8. There's some outlier species like zebras, who for some reason cannot be predicted with this clock. I'm not sure why, but most species are really highly predictable. Although this just uses one single biological sample and everything else is just noise. really showing aging clocks. They are readouts of something downstream. They are readouts of how noisy your system gets, how less well regulated it gets. But the question is, of course, what are the drivers of a stochastic variation? Where does it come from? Why does it happen? And this is something we wrote in a perspective. If you want to read these, I can, of course, also share these with you. Otherwise, you can also scan the QR code here. And here we wanted to have a bit deeper dive into what the stochastic variation is and where it's coming from. And here we need to understand that biological systems are inherently stochastic. It starts with, oops, sorry. Okay, here you go. It starts with Brownian motion. So every single molecule in your cell is behaving randomly with some constraints. Then you have stochastic DNA damage. It's happening every second in every of your cells. Usually it's, of course, faithfully repaired, but sometimes you have stochastic somatic mutations, and sometimes you just have incomplete repair. It might just not get repaired. You have stochastic epi mutations, so DNA methylation or histone modifications that change, maybe during replication, maybe during DNA repair, maybe just randomly. Essentially, every time it has to be copied or repaired, there can be mistakes because no maintenance system is perfect. Transcription is noisy. You have even two types of noise, the extrinsic noise and the intrinsic noise. Extrinsic just meaning the signals that come from the outside, environmental factors, temperature, hormones, and so on. Intrinsic noise is just how the machinery works, because transcription works in bursts, and this is a noisy process. Translation, of course, is a stochastic process. You have here, for example, the stoichiometric balance of the ribosomes. That needs ideally to be perfect, but it's not always. The abundance of ribosomes changes. You have tRNA abundance that might not always be perfect. And then protein folding, of course, is not a perfect process. So you can have problems and mistakes at every step. And this all leads to a degradation in the network. So in young and healthy individuals, you have a gene regulatory network that is properly functioning. Gene A is properly influencing gene B, for example. But now with age or disease, this might break down due to all these different small mistakes that happen every day in your cells. Some connections might get weaker, some might get stronger, whereas others might be completely lost. So this is essentially a spiral that gets worse and worse, because with weakening network stability, of course, your maintenance is also behaving worse, and you might accumulate even more mistakes even faster. And the stochasticity is hierarchical. So if you have DNA damage, this needs to be repaired. But you repair, as I mentioned before, you need to copy the epigenome as well as the genome, of course. But because there's no perfect maintenance, sometimes there are mistakes. And now suddenly, although this should be methylated, it's not methylated anymore. The same can also happen during mismatch, maybe also during replication. If you have a mismatched base, then maybe the methyltransferase, DNMT1, mistakes this mismatched base as a methylated C, and suddenly it methylates the sites. The base itself is repaired, there's no problem there, but suddenly you have a methylated base, although it's not supposed to be methylated. And then, of course, you can have irreversible loss of methylation due to a somatic mutation. So if the base itself mutates, then of course it cannot be methylated anymore. And this might be then irreversible. And this can trickle down. So if you have a gene that is repressed, so the promoter is 100% methylated, the gene is completely inactive, then the only way a random process can go is to less methylation, because it's already 100% methylated. So if there are any mistakes happening during repair, replication, maintenance, at some point something will happen and it's not methylated anymore. Initially, maybe it doesn't do anything if there's just one small mistake, but again, you have more methylated states than non-methylated. And just by pure chance, it might happen that, again, something happens and it's even less methylated. And at some point, the gene is not repressed anymore. So they get de-repressed, they get activated again. Maybe not to a full level, but suddenly you still have a sporadic gene expression happening. And this is exactly what we see with aging. Suddenly we do see developmental genes active again. If you look at a liver cell, you suddenly see genes expressed from different cell types that are not supposed to be there. You have a loss of cell type identity. Some people interpret this as there might be a program, maybe the developmental genes continue running into aging and adulthood. We think this is really just due to imperfect maintenance, because these genes are supposed to be completely shut down, repressed, but then due to imperfect maintenance, these genes get activated again. So it's compatible with aging. And now lastly, maybe a short conclusion. The chronological age can almost perfectly be predicted, but we lose biological information. Aging clocks are confounded by cell type composition. And the real underlying explanation of aging clocks is increasing stochasticity. And now I just want to spend a few minutes on how aging clock could be useful in the future. And I think one big point is patient stratification. So we might identify individuals that are at risk before a disease breaks out. So we can go from a reactive therapy to a prospective risk assessment. And here's just one collaboration with the company. So I was a consultant for this company. I have to declare that. I don't receive money for this though. And we developed an aging clock based on clinical laboratory profiles. In the end, we just measure 6 measurements. It's albumin, creatinine, the HbA1c, ALT, HDL, and triglycerides. And we can highly accurately predict the biological age. So these are standardly measured measurements at your doctor, of course. And we validated this in different data sets, and it seems to work fairly well across different populations. We do see that it really nicely correlates with the chronological age. And interestingly, it does associate with diseases. So we do see, for example, chronic kidney disease is almost 10 years predicted older. Diabetes as well, chronic heart disease, and several other diseases are predicted significantly older than healthy individuals. And maybe even more interestingly, you can see an effect already before the disease is diagnosed. So here, this is, for example, for type 2 diabetes. At the age here of the relative years of 0, the diagnosis was done. So this is longitudinal follow-up data. But we could already 5 years before the disease was identified see that these individuals have an accelerated aging. So these individuals are 2 years older than healthy people, although they are not even diagnosed with diabetes or kidney disease, for example. So up to 5 years before diagnosis happens, we already see an elevated age. We only see the elevated age, so we only know this person is 2 years or 5 years older than they should be. We don't know which disease might happen, but they have an increased risk of mortality and diseases, which then can help to say, okay, this individual is at risk, the doctor should look more closely, maybe every half a year follow-up, or maybe even more specific tests to see whether there's anything that might be a problem. Now, there are, of course, even more developments. One really promising one is plasma proteomes. This is really expensive, but it has been shown in several publications now that you can, from one blood draw, identify proteins that are associated with different organs. So you might, with one blood draw, might be able to identify whether your kidneys are older than they should be, or your heart, or whether several organs are too old. So with this, you can even pinpoint it even better. You draw blood, you do this aging clock prediction, and you can say, okay, your kidney is older than it should be. We have to do something here. This is, of course, super expensive and still in development. It's not a clinical tool, but this shows where it could go in the future. So just to conclude this part, and then also at the end of my presentation, clinical blood biomarkers can already be used to predict biological age and disease risk. They can be used for patient stratification. And I think it's also interesting though for this group, because an African clinical aging clock could really be a great study for Afrolongevity. Because so far, to my knowledge, this has really not been done. And we also don't know whether these aging clocks would even work in African populations, because it is known that many of these biomarkers have differences in different populations and ethnicities. So maybe the clinical blood profiles are shifted in such a way that the clock just doesn't perform. And this, of course, also is true for DNA methylation clocks and transcriptome clocks and proteome clocks and so on. And there's just currently not sufficient data to do this. And I think this would be a great study if there's any collaboration with a hospital where one could get such data, of course, with an ethics approval, that one could try to build such an aging clock and see how well does it perform, and can this be something African specific even. So with this, I'm at the end. I would like to thank your attention, and I would be happy to take your questions. Thank you. I will stop sharing. It's a bit easier to see the chat as well. Just have to get my charger, my battery. So yeah, I think there are several difficult topics. Especially stochastic part is not easy to understand. So it's I wouldn't be surprised if there are many questions for that part. But you can of course also write me afterwards if you have anything that you would like to discuss. If you have any questions that come up later.
Okay, thank you so much, Dr. Meyer, for that insightful class. I hope there are some clinicians that we send in some questions. They are not in the class now because they informed me ahead of time because of the schedule, and I think they'll listen to the recorded versions and ask questions. Meantime, if you have any question, please do raise your hand and then ask your question. Thank you. Okay, I'll go first. Um, this is a question that I think would help us think well and brainstorm properly with regards to the condition of Africa. Africa is completely different from other population. And because for over the years, past years, or based on our historical dimension, we are divided along ethnic lines, and what affects like the weather of those in West Africa is completely different for those in South Africa. So here, for example, we have the rainy season, dry season, and all that. Then you go to Southern Africa, you have the winter, summer, autumn, just like those in most Europe and America. But then they also have a different disposition, you know. So if one is to develop an aging clock, are we going to call it African aging clock, or are we going to design it based on these geographical differences?
Yes, this is an excellent question. So short answer is, I guess, we won't know before we try. I think there will definitely be differences in different regions. I mean, the clock I showed you in the end, we built it on the Southeast Asian population, largely from Thailand. And we checked it in an US and European population as well. And there seems to perform quite well. There are slight shifts in the predicted age. So the Americans, for example, are generally predicted a bit older. But it's all within 1 year or so. So it's not a big shift. But there are also other publications for the aging clock showing quite strong differences. Some even then don't show any correlation to age anymore. So there can be really dramatic differences depending on what clock you're looking at. And of course, to my knowledge, no clock has been really tested in a large variety of African populations. And we do know that genetics, of course, plays a big role. So depending on genetic setup, the aging process might be somewhat different. But of course, also the environment. So temperatures plays a role, the disease risks and so on, of course, food. So everything plays into this, and this is really complex. But I also think it's a really great benefit because there is so much diversity, much more than we have in other countries or continents even. So I think there's a lot to understand what is actually be robustly associated or maybe even driving aging. There's, for example, one really nice study from, I think, 1 or 2 years ago from Alan Cohen, where he showed that in Western populations, we have a lot of inflammation with aging. But then he looked at some populations from, I can't remember where it was, some remote island, I think. And there, they did not find any evidence for inflammation with aging. So it seems to be something maybe either due to the genetic setup or to the environment, the diet and so on, that drive this inflammation. So it might not be universal. And maybe there are still several other things that we think are universal to human aging, but maybe that's completely specific to the populations we study because we just don't know. And this is why I think it's so important to have a look. Maybe first of all, of course, because the tools that we're using at the moment, the aging clocks, the disease risk predictions and so on, they are just not properly validated on African populations. Almost everything is validated on Western populations from the US or maybe Europe, but we don't know how well they will perform at all in other populations, including Africa. And this is, of course, a big problem because then it cannot be applied to these populations. And it also means that we don't understand the underlying signal, of course. So we need really to see what is universal and what is maybe affected by the environment, what is affected by genes, or what is affected by the interaction between those two. It's, of course, also one thing that might be important. And without this data and without this analysis, we just won't know.
Okay. Thank you so much. If you are there other questions, let me check the comments section. There's no question. I think they're going to struggle a bit with the to make sense of the presentation, but please do send me the slides. I will send it across. They might have questions. They'll reach out to you via email. And some of those who also are joining through YouTube will be able to make sense of the presentation better. Yeah.
Yep, don't hesitate to reach out. It's a complex topic, so don't worry about your questions also now or later on.
Okay. Ambassador Somini, your final words, please. You are able to talk.
Thank you so much for the session. And Dr. David Meyer, thank you so much for making time to present. That was a very thought-provoking presentation. I personally learned a lot. And I know that those of us who are live here, we can all attest to it. And so we want to thank you all for joining us. And we hope to see you the next class session. And thank you.
Yeah, thank you for having me on board.
Thank you so much. All the best. Bye for now.
Okay, see you around. Bye.